Dear students, today let us discuss on the introduction to the application of X-ray diffraction and mass spectrometry. The functional properties of biomacromolecules are closely related to their complex three-dimensional structure as well as their molecular mass. Therefore, understanding the detailed 3D structure and mass of biomacromolecules such as protein, nucleic acid, etc. is highly required. X-ray diffraction analysis is used for derivation of three-dimensional structure, and mass spectrometry is important technique for determination of mass of a biomacromolecules. These two techniques are important in entire field of biochemistry because they have been given an insight into how proteins function as enzymes, how they fall and how they evolve, and how proteins recognize and bind other molecules. Now, X-rays. X-rays are very high energetic electromagnetic radiation. It has short wavelength ranging from 0.1 to 1 nanometer and frequencies ranging from 3 into 10 to the power 16 to 3 into 10 to the power 19 Hz. Due to the shorter wavelength and high frequency, it has high energy in the range of 100 electron volt to 100 kilo electron volt. X-rays were first observed by Wilhelm Conrad Roentgen, a German scientist, in the year 1985. He found them accidentally when experimenting with vacuum tubes. X-ray can be generated by an X-ray tube or a vacuum tube. Now, let us know the crystal. A crystal can be defined as regularly repeating three-dimensional array of atoms or molecules. The table salt, graphite, diamond, and quartz are examples of natural occurring crystals. Application of X-ray diffraction analysis. X-ray diffraction or X-ray crystallography is used in the structural determination of biomacromolecules such as protein, nucleic acid, etc. X-ray diffraction is ideally suited for soluble protein but less appropriate for ribosome, proteasome, and membrane proteins which are difficult to crystallize. Now, let us have a brief discussion on the X-ray diffraction analysis of protein biomacromolecules. The first step in the structural determination by X-ray crystallography is the crystallization of the protein. This is the most important and laborious step. Some proteins are crystallized by addition of salt such as ammonium sulfate, that is called a salting out process. It favors the formation of highly ordered crystal as it reduces the solubility of proteins. For example, myoglobin crystallized in 3 molar ammonium sulfate. Some proteins crystallize readily, whereas others do so only after much effort has been expended in identifying the right conditions. Importantly, the protein crystal have a series of regular plane known as lattice plane. Once the protein is crystallized, it is placed in an X-ray beam between X-ray source and a detector. The reason behind the use of X-ray in determination of three-dimensional structure of biomacromolecules is that their wavelength range is in the same order of magnitude as chemical bond does, allowing us to see a resolution equivalent to interatomic distance, that is means 0.8 to 2.5 angstrom. Atomic planes of a crystal causes an incident beam of X-ray to interfere with one another as they leave the crystal. This phenomenon is called X-ray diffraction. In 1913, Braggs proposed that a crystal may be regarded as a series of planes which behave as a mirror, reflecting X-rays. A crystal scatters an incident X-ray beam in a huge number of different directions. Diffraction can result in interference between diffracted waves, which result in the wave either reinforcing or weakening each other, depending on their relative phases. These phenomena are characteristic of wave function and are known as constructive when the waves are in the same phase, and the destructive interference when the waves are in the different phase respectively. From this idea, Bragg's law was derived an equation n lambda is equal to 2d sin theta, where d is the distance between the atoms in the parallel crystal plane, theta is the angle of reflection, and lambda is the wavelength of the X-ray, and n is the integer, either it may be 1, 2 or 3 and so on. The only experimental variable in the Bragg's equation is the angle of incidence theta. Variation of theta is achieved by a slight rotation of the crystal approximately 1 degree between exposure to the X-ray beam, allowing detection of distinct pattern of Bragg's reflection for each angle of rotation. In a complete data set, most atoms in the crystal lattice will contribute to some diffraction patterns. The scattered or diffracted X-rays are detected on an X-ray film or solid-state electronic detector, where the brightening emulsion is proportional to the intensity of the scatter beam. These intensities and their position of its spot are basic experimental data of an X-ray crystallographic analysis. Therefore, intensity of its spot is measured and recorded. The next step is to reconstruct an image of the protein from the observed intensities. Mass spectrometry. Mass spectrometry is an analytical technique that accurately measures the mass of a sample, which can be a single atom or large molecule, for example protein. In this technique, samples are firstly ionized in the gas phase by different methods and electrostatically directed into a mass analyzer based on their mass to charge ratio and finally detected. Thus, mass spectrometry has three basic components, namely ionization or ion source, the mass analyzer, and a detector. Now, the application of mass spectrometry. In early period, mass spectrometry was primarily used for determination of atomic mass of an element and natural isotopes. Application of mass spectrometry in biological systems started in 1940s when heavy stable isotopes were used as a tracer to study the carbon dioxide production in animals. With improvement in the technique, mass spectrometry made possible to determine the mass of volatile and non-volatile compound. However, it could not apply to the macromolecule such as protein and nucleic acid. This problem was overcome by introduction of two methods such as electrospray ionization technique developed by Fan and his co-worker in the year 1988. Number 2, matrix-assisted laser desorption or ionization technique by German scientist Helene Kurve and his co-worker in 1988, and a Japanese scientist Tanaka and his co-worker in the same year. For this pioneering work on development of soft desorption ionization methods for mass spectrometric analysis of biological macromolecule, John Fan and Koichi Tanaka were awarded Nobel Prize in Chemistry in the year 2002. Application of mass spectrometry in biomacromolecules. Number 1. Matrix-assisted laser desorption or ionization time of flight mass spectrometry, simply MALDI-TOF-MS, and electrospray ionization mass spectrometry or ESIMS are used in accurate determination of molecular mass of protein in pure as well as a mixture of proteins. Number 2. Tandem mass spectrometry or MS2 is used in the peptide mass fingerprinting to sequence short states of polypeptides. Using isotope-coded affinity tags ICAT, MS is used for quantification of individual proteins in a complex mixture. Therefore, mass spectrometry can be used for simultaneous comparison of expression level of all proteins in cells grown in different media. Mass spectrometry is conveniently used to detect the change in the mass of macromolecule due to the presence of bound cofactor, bound metal ions, covalent modification, etc. Now let us discuss how the mass spectrometry is used to determine the accurate mass of biological macromolecule. A. Multi-top MS. Matrix assisted laser desorption ionization time of flight mass spectrometry has been successfully used to measure the mass of a wide range of protein. In this technique, proteins are co-crystallized with the light-absorbing organic matrix such as cyanogenic acid or alpha-cyano-4-hydroxycyanamic acid. The matrix is then applied to the ceramic or metal plate and allowed to dry. When a short pulse of laser light, means high intensity, is applied on the plate, the proteins are ionized and dissolved from the matrix into the vacuum system, which is accelerated in an electric field and fly towards a detector. As they travel through the flight tube, the smallest protein molecule move faster and arriving at the detector first in compared to the bigger protein. Thus, the time of flight in the electrical field is a measure of the mass, or more precisely the mass to charge ratio. The method is highly sensitive, therefore can be analyzed as small as few picomoles to femtomoles of biomacromolecules. So, the protein that are extracted from the two-dimensional electrophoretic gel can be applied. B. Electrospray ionization mass spectrometry or ESIMS. In this method, the protein solution is passing through a needle that is kept at a high electrical potential. The protein is fully charged, therefore dispersing into a fine spray of charged microdroplets. As they pass through several stages of decreasing pressure or sometimes high temperature, the solvent surrounding the macromolecules rapidly evaporates, and the resulting charged macromolecular ions are thus introduced non-destructively into the gas phase analyzer and measure the mass of the protein. Number C. Tandem mass spectrometry is used in the peptide fingerprinting. In this technique, the unknown sample of interest is extracted and specifically cleaved into smaller fragments by a chemical or enzymatic process. The mixture is then injected into a device that is essentially two mass spectrometers in a tandem. In the first, peptide mixture is sorted so that only one type is selected and further fragmented in a chamber between the two mass spectrometers, and then fragments are sequentially ejected and determine the masses by second mass spectrometer such as multi-top or ESI-top. Finally, the peptide masses or fingerprint are compared against the fingerprint found in the database of non-protein sequences or even the genome that have been electronically cleaved by a computer simulating for the same fragmentation technique used by the experimental sample. Now, conclusion. As we know, biomacromolecules such as protein and nucleic acid can be crystallized and therefore possible to determine the three-dimensional structure of these molecules by X-ray diffraction analysis. Different methods of mass spectrometry such as multi-top MS and the ESIMS are important techniques that use for determination of precise masses of biomacromolecules. Tandem mass spectrometry is used for the sequence of short stretches of polypeptide or peptide mass fingerprinting and the quantification of individual protein in a complex mixture. Therefore, mass spectrometry can be possible to simultaneous comparison of expression level of all proteins in cells grown in different media. Mass spectrometry is also conveniently used to detect the changes in the mass of macromolecule due to the presence of bound cofactors, bound metal ions, covalent modification, etc.